Welcome back, students. Today we are going to discuss about replication of DNA. Already you have studied what is DNA and the replication of DNA. Everything you have studied in your lower classes. And today we are going to discuss about the replication of DNA. We are supposed to study the replication of DNA, that is the general process, and also the replication of DNA in prokaryotes and also eukaryotes. Okay. What is replication of DNA? You all know the definition. What is replication of DNA? It's the process in which DNA copies itself to produce identical daughter molecules of DNA. What is replication? Replication is the process in which DNA copies itself. Which one is copying? DNA is copying itself to produce identical daughter molecules. DNA is copying itself to produce identical daughter molecules DNA. Clear? What is replication? Replication is the process by which DNA copies itself to produce identical daughter molecules of DNA. And coming to the DNA replication, the general processes, we all know which one is the genetic material. DNA is the genetic material. Once the DNA has to duplicate and divide, it should copy the organelles and biomolecules. So the biomolecules and the organelles should be copied and it should be distributed among all cells. We know DNA is the genetic material. If it has to duplicate and divide into daughter cells, we know it is dividing. In DNA replication, it is dividing. Once it has to divide and duplicate, what it has to do? It has to copy the organelles and the biomolecules and it will be distributed among the cells. And we know there are 4 nucleotides. Which are the 4 nucleotides? Adenine, guanine, thymine and cytosine. And an adenine will be pairing with which one? Adenine will be pairing with thymine and guanine will be pairing with cytosine. These basic things, you know, there are 4 nucleotides which takes part in the DNA replication process. That is adenine, guanine, thymine and cytosine. That is, DNA is made up of 4 nucleotides and these are the pyrimidine bases. Okay. And we know the DNA is made up of 2 strands and they'll be forming a ladder-like structure, and at some points they'll be forming a twist. It is ladder-like structure, and they will be binding with the help of hydrogen bonds. And at some points they'll be forming a twist, and because of this twist, it will be looking like a double helix structure. And the nucleotides will be bind together by hydrogen bonds. Okay, DNA is made up of 2 strands. That is, they are formed like a ladder-like structure, and the bonds between the nucleotides is the hydrogen bonding. And binding of adenine with thymine and guanine with cytosine, will be calling it as the base pairing. And in some places, some areas, it will be the nucleotides will be pairing up and there will be a twist formed, and because of that you are having a double helical structure. Why this DNA is having a double helical structure is the nucleotides pairing up will be forming a twist, and because of that twist we are getting the double helical structure. And we know that the 2 strands we are designating as the 5' end and the 3' end. Why we are calling it as the 5' and the 3' end? 5' and 3' represent the 5th and 3rd carbon of the sugar ring. Okay, carbon atom of the sugar ring. The strands we are designating as 5' and 3'. What is this 5' and 3'? They are the 5th and 3rd carbon atom of the sugar ring. And this 3' end is very important because the nucleotides will be added to this end. Okay, so we have 2 strands. The end strands we are designating as 5' end and the 3' ends. The 5 and 3 represents the position of the carbon atom of the sugar ring. And which end is important? The 3' end is important because during replication, new nucleotides will be added up there. And while studying the process of replication, there are three main steps, that is initiation, elongation and termination. What are the three major steps in the replication process? Three steps: initiation, elongation and termination. So first is the initiation. Under initiation, we will be studying the terms like origin of replication, the role of helicase enzyme, SSB protein, that is single strand binding protein, and also DNA primase enzyme. So what is this origin of replication? From the term itself we can tell the point at which the replication begins is known as the origin of replication. What is origin of replication? The point at which replication begins is called as the origin of replication. Next is the helicase enzyme. What is the role of this helicase enzyme? It is an enzyme which separate the two strands. So what's the role of helicase enzyme? Helicase enzyme will be bringing about the strand separation, and this leads to the formation of the replication fork. What this helicase enzyme will do is it will be breaking the hydrogen bond between the base pair, and ATP is utilized for the purpose. What is origin of replication? The point at which replication starts is called as the origin of replication. What's the role of this helicase enzyme? Its role is to break the two strands. Break the strands, and it leads to the formation of replication fork. It will be breaking the hydrogen bonds between the base pair, which is energy is utilized, and we'll be getting the energy from ATP. Next is single strand binding protein. Actually, this single strand binding protein will be binding to the single stranded DNA, and it will be stopping the strands from binding again. Okay, helicases will be forming the strand separation. The single strand binding protein will be binding to a single stranded DNA, and it will be stop the strands from binding again. Stop the strands from binding again. Next is the enzyme DNA primase. Actually, for DNA replication, we need short fragments of RNA, that is primer is needed. The short fragments of RNA, the RNA primer is formed with the help of the enzyme DNA primase. A primer is required. The primer is the short sequence of RNA. It is around 10 nucleotides in length, and for synthesis of this RNA primer, which one is needed? DNA primase is needed. So for the DNA replication, an primer is needed. Primer is short sequence of RNA around how many nucleotides in length? 10 nucleotides in length, and DNA primase enzyme will be synthesizing the RNA primers. Okay, so once the strands are separated, the replication can be initiated. For the initiation of replication, RNA primer is needed. RNA primer is synthesized with the help of DNA primase. This is regarding initiation. Initiation is taken at that side. The point where initiation takes place is the origin of replication. Then helicase enzyme will be separate the two strand, and the single strand binding protein will prevent the joining of the two strands. Again, we have, for starting the DNA replication, we need short fragments of RNA, short sequence of RNA, which is around 10 nucleotides in length. That is called as synthesized with the help of the DNA primase enzyme. Okay, what's the second step? Second step is the elongation. Elongation is done with the help of the DNA polymerase 3 enzyme. Which enzyme will bring about the elongation? Elongation step will be done with the help of the DNA polymerase enzyme, and this will be forming the new strand. It has the capacity to read the nucleotide in the template strand, and it will be forming the new strand. So it will be make the new strand. How it will be making the new strand? By reading the nucleotides present in the template strand, it will be forming the new strand. If the In the template strand, if adenine is present, it will be add the thymine to the incoming strand. Likewise, it will be forming the new strand of DNA, and it can synthesize strands in the direction of 5' to 3' direction. And if you see, we know we have two strands, that is, one is the lagging strand, another one is the leading strand. In the leading strand, the synthesis of DNA is continuous, whereas in the lagging strand it is not continuous, and small pieces, that is Okazaki fragments, will be formed. In the lagging strand, the DNA will be synthesized as fragments. The fragmented DNA, we will be calling it as the Okazaki fragments. Okay. Next is this termination. So how elongation occurs? Elongation will be occurring with the help of the enzyme DNA polymerase 3 enzyme. It will be reading the nucleotides present in the template strand, and it will be adding the complementary base pairs. That is, for adenine it will be pairing with the thymine, and it can synthesize only in 5' to 3', and we have two strands. One is the which are the two strands, leading strand and the lagging strand. In the leading strand, the synthesis of DNA is continuous, whereas in the lagging strand, the synthesis is discontinuous. Small fragments of DNA will be produced, and that fragments of DNA is called as the Okazaki fragments. Okay. Next is termination. Finally, our initiation step over, elongation step over, and final step is the termination. And in the final step, the RNA primer which has been initially added for initiation will be removed. It will be removed with the help of DNA polymerase 1. Even RNase H also will help to remove the RNA primer. RNA primer, for the initiation of replication, we need small fragments of RNA, that is called as the RNA primer, that will be removed with the help of DNA polymerase 1 and also RNase H. Next is the DNA ligase enzyme. What it will be doing is, we have studied in the lagging strand, the formation of DNA is discontinuous. There will be gap in between, and we will be forming small fragments of DNA, that is called as the Okazaki fragments, that will be joined with the help of this DNA ligase enzyme. So these gaps are filled with the help of this DNA ligase, and it closes the nicks in double-stranded DNA. Okay, fine. And finally, the replication process will be complete when all the primers are removed and ligase is filled in the gaps, and this process will be getting two identical sets of genes, and this will be passed on to two daughter cells. Okay. So at the end of the replication process, we will be getting two identical set of genes which will be passed on to the next to the two daughter cells, and the entire cell division process will be completed in 1 hour. So I hope you know the steps. We have initiation, elongation, and also termination. Mainly the important things you should know is we have two strands, leading strand and the lagging strand. In the lagging strand, we have which one? Okazaki fragments, and this is the DNA polymerase 3 enzyme which helps in the formation of DNA. Then helicase enzyme, it will be forming the separation. It is strand separation. Single strand binding protein prevent the joining of the strands. Then the short fragments of DNA formed is called as the Okazaki fragments, and this DNA polymerase 1 will help in the removal of the RNA primer, and DNA ligase will help in the joining of the Okazaki fragments. So this diagram we can represent, and the point where replication takes place is called as the origin of replication. So this is the two strands of DNA, and with this the general steps for DNA replication is over. I hope you are clear. Okay.